Therapy Association, we really appreciate your being here. And also, really appreciate the speaker who I'm going to introduce in just a moment. We're very, very fortunate to have our hometown expert of this kind of stature and humanity. But before we make that introduction, my name is Dennis Tersch. I'm the president of the New York City Cognitive Behavioral Therapy Association. It's something we've been doing for the last 6 years or so. It's been a growing organization, and we really had a mission to create a community of scientists, practitioners here, where people who have a commitment to research and evidence-based practice, and also a commitment to delivering best practices of clinical services, could find each other and build a community. So these events are very important for them to that aim. Also, at the ABCT convention, the Association for Behavior and Cognitive Therapists, or Therapies, or Therapy, depending on, I guess it's America, so it's therapy, right? In England, they like to say therapies for these kind of organizations. So we're going to have a get-together at ABCT in the, appropriately in the hotel bar, at the 720 South Bar and Grill, and it'll be between 5 and 6 on Thursday, November 12th. So if you're there, it'll be great to see you. And we have two other speakers who are coming up within this academic year. We'll have Marv Goldfried here in February, and Sabine Wilhelm here in May, speaking on relationships and case conceptualization, Marv's case, and also speaking, Sabine's going to be bringing her expertise in the treatment of OCD and anxiety disorders, so hopefully we can see you there. So I'm not a big fan of reading elements of someone's bio off of pieces of paper, off of phones, but in this case I've made an exception because Dr. LeDoux is truly an accomplished neuroscientist, and not just on the basis of his research, but on the way that he's translated very complicated ideas from the sciences into human terms, applicable terms, so that they reach people and they inform people's lives and they can inform the way we do therapy. There have been many clients over the years who have mentioned, sort of unprovoked your work, and they've been moved by it. So it's something that I appreciate in a really honest, authentic way, actually. Dr. LeDoux is truly one of the foremost neuroscientists in the world today, and his research explores the biology, the biological underpinnings of emotion, of awareness, of threat perception. He's the Henry and Lucy Moses Professor of Science at NYU and in the Center for Neuroscience, and he directs the Emotional Brain Institute of NYU and the Nathan Klein Institute. He's also a professor of psychiatry and child and adolescent psychiatry at NYU Langone Medical School. His work is focused on the brain mechanisms of memory and emotion. And I highly recommend, if you haven't, and many of you have, reading his wonderful books, The Emotional Brain, Synaptic, Self, and Anxious. He's received numerous awards, including the Carl Spencer Lashley Award from the American Philosophical Society, a Fitson International Prize in Cognitive Science, American Psychological Association Distinguished Scientific Contributions Award, and many, many other honors. And every moment that I spend further expounding on these awards is one less moment we'll have to actually hear the man talk about the wonderful things he's here to talk about. So without further ado, here's Dr. LeDoux.
Thank you, Dennis, and thank you all for coming. Let's see if this thing is going to cooperate now. Okay, great. So, tonight we're going to be talking about fear and anxiety, mostly fear, I guess, but also anxiety. And a lot of what I'll be talking about is from my new book, which is called Anxious. And some of the themes that will come up is, what we can and can't learn about fear and anxiety from animal research, why drugs are not more effective, and how psychotherapy might be improved by research on the brain, including the brains of rats, which is what I study. You may be suspicious of that statement, but I hope by the end you'll see how it's relevant. So, what do we mean by fear and anxiety? I mean, everybody kind of has their own definition, so I'll tell you what I mean. So fear is a situation where a threat is present, whereas anxiety is a worry or an anticipation of a threat about some future source of harm that may or may not happen, may not even be physically plausible. A person who is anxious about alien abduction is just as legitimately anxious in a sense. I mean, their anxiety is just as real to them as a person who's anxious about their mortgage payment. It doesn't have to be something that can actually happen. So, I'm going to be talking about all this from the perspective of the brain. As a neuroscientist, I sort of, I drink the neuroscience Kool-Aid, and I think all aspects of mental life and behavior are products of the brain. That doesn't mean that we can explain everything we need to know about the mind in terms of the brain, and we may never be able to, but somehow the brain is necessary and responsible for all of these things, no matter how mysterious they are. So when we're looking at the brain, we have to think about three different levels of explanation. One is the level of behavior, which is what we can measure in a human or another organism. In people, we have the advantage, we can also use their verbal behavior as an output. But you have to have something to measure in order to relate it to the brain. And in fact, your ability to understand some psychological process in the brain is only as good as your measurement tool and its validity in accessing or assessing that function in the brain itself. And then once we go into the brain, we have to understand the level of brain areas and brain systems. Brain areas, we often talk about the frontal cortex or the hippocampus or the amygdala as if these actually do something, but they don't do anything on their own. They only perform functions because they're parts of systems, because they have circuits that connect with other parts of the brain and so forth. So we like to talk about systems rather than areas, even though I will talk about areas because they are part of systems. And then there's also the level of cells and molecules, and we have to get to this level as well, because if we want to, if you believe that drugs may potentially be useful in treating people, this is the level we have to understand how drugs work, so that we can then take them back up to the level of behavior. Now, normally you might think of a neuroscientist as taking a reductive approach. The idea is to get finer and finer and finer explanations until you're down to the level of the gene. But that's not really the way I work, and I think, a lot of neuroscientists don't think in those terms either. We're more likely to think in terms of integration across levels. So we aren't necessarily trying to simply get to the gene and say, okay, well, that's the answer. Findings about genes can actually inform us about behavior. So, a gene may be able to separate some behavioral task into components that the gene is involved in and not involved in, and give us new information about behavior in the circuits and systems that are responsible. Same thing with brain systems. The whole idea of short-term memory being physiologically different from long-term memory came from the famous patient H.M., who had, after hippocampal surgery for epilepsy, had intact short-term memory, but wasn't able to convert that into a long-term memory. That meant that the lesion of the hippocampus disrupted his ability to form new long-term memories, but didn't interfere with his ability to hold information in mind for a short period of time. So the brain can tell us about things. So studying the brain can reveal dissociations in behavior. Studying cells, molecules, and genes can also dissociate behavior. But behavioral finesse, our ability to understand behavior, is critical in order to get to the finest explanation at the level of the genes, which can then feed back up. So we have to start with a good understanding of what it is we're studying at the behavioral level before we can proceed on this stuff. Now, as I said, I'm going to mainly be talking about fear, because that's the general area that I've worked on most, and I think it's easier to study in animal models than anxiety. So how can we study these things like fear and anxiety in animals? So even though I don't study anxiety in animals so much, it is done quite a bit. So we start from animal behavior, and these were some of the pioneers in this field, names that you're probably all familiar with. And the studies of animal behavior was the way that Skinner and Volpe and others began to do behavioral therapy by taking principles such as extinction from these behavioral studies in animals, applying them not only to human behavior but also to clinical treatments. And then, of course, cognitive behavioral therapy is an extension of behavior, and then cognitive therapy of cognitive behavioral therapy. I'm not an expert in all this, so if I have some of it wrong, please correct me. But we start with animal behavior, and then we can get to these points. I think the relation of cognitive therapy to animal behavior is probably the least dramatic. But I think there are some important connections that will come up later. So the easiest thing to study, and I think one of the most important things to study in animals, is Pavlovian conditioning. Why? Because it's really so important in our lives. All of the stimuli that are triggers in our lives for good things and bad emotions and good emotions really are based on associations that we form between different stimuli as we go through the world. Something bad happens to you on the street corner in New York, that street corner becomes a trigger. Maybe street corners in general become a trigger. If it was at night, maybe nighttime becomes something that makes the person have problems. These are all learned through associative processes. And I've studied this kind of simple thing where you give a rat a tone, pair it with a shock, then a few hours, days later, you give the tone again, and if the animal had the tone and shock paired, he will freeze, his blood pressure and heart rate will go up, stress hormones will be released. Same thing that basically happens in a human that encounters a threat. They'll have some muscle tension, blood pressure and heart rate are moving blood around in the body to get nutrients to and energy sources to the muscles that are needed for this fight-flight kind of response. And hormones are being released as well. So we can study these things in animals, not because rats and people are afraid of the same thing, but because a threat is a threat in a sense. And the threat will go into the brain, activate certain systems in the brain, and produce these responses which are very similar in people and animals. So over the past 30 years, we've learned a lot about the behavior. We've got tons of information about the neural circuits that are involved, a lot of information about the cells and the molecules that underlie this kind of learning process. Now, let's just walk through a little bit of this here. So what happens is that the tone is going to come into the brain, reaching the auditory thalamus, which the usual way of thinking about it is going to send information to the auditory cortex, and that information can then reach the amygdala, which is a big part of what I'll be talking about tonight. And there's a lot of circuitry in the amygdala that we'll talk about. But in addition to that pathway from the auditory cortex, there's also a pathway directly from the sensory areas of the thalamus, visual, auditory, and so forth, into the lateral amygdala. So the thalamus can turn the amygdala on actually before the cortex has begun to process the stimulus, or at least at the same time, and can begin to trigger these responses, defensive behavior, autonomic nervous system responses, hormonal responses, before you consciously know what it is that is out there. As the information goes through sensory cortex onto higher order cortical areas, especially the prefrontal cortex, you become consciously aware of what the stimulus is. But by the time you're consciously aware of it, your brain has already been triggering all these responses because these are all innate responses that you have a very fast pathway here. Even from the sensory cortex, you're triggering responses before you know what it is that you're responding to by way of the prefrontal cortex. So the lateral amygdala is the sensory gateway into the amygdala. The central nucleus is the floodgates of responsivity. You open up the central amygdala, and you get all these different kinds of responses. The central amygdala also arouses the brain, which is very important in anxiety disorders where you have hyperarousal, and we can talk about that later. And then all these molecules, we're not going to be talking about that much tonight. So why has it been such a useful task? Well, many of the things that elicit fear in humans do so because of associative learning, as I've said. Associative learning in so-called fear conditioning is rapid, and the memories are persistent. The responses are not learned, but are hardwired. What gets learned is the meaning of the stimulus. So a novel stimulus now gets to elicit these hardwired responses. So even though we call these things conditioned responses, the responses aren't conditioned. The more appropriate term is conditional responses. They're conditional upon the novel stimulus having been associated with something else. Many kinds of stimuli can be conditioned this way. It works similarly in rats, humans, and many other animals. And so identifying the mechanisms in rats helps us understand human fear. All right, so in terms of mental health research, we have this fear conditioning procedure has been used in trait anxiety, panic disorder, PTSD, psychopathy. Somebody asked me about psychopathy, and here's the only thing I can tell you about that is in this reference here. Criminal behavior, psychiatric symptoms in active duty Marines, early life vulnerability to psychiatric problems, and on and on. So it's a very, very popular task that's widely used because of its ability to trigger certain kinds of responses in the brain. For example, to activate the amygdala and see if, for example, people who've been stressed in early life have a more active amygdala than people who haven't. I'm not saying they do or don't, but that's one way you could use this. So in many ways, this procedure and all of the research that's been done on it, which we started about, as I said, about 30 years ago, has been a resounding success. But here we are at a crossroads now. We could stay the course, keep doing things the same old way. I think more discoveries, maybe even more important ones, will result. Or we could ask, what is it that we've been doing when we do this kind of fear conditioning research? And might we even do better if we had a sharper conception of what we're studying? As I said at the beginning, our ability to study the brain mechanisms of something is only as good as our ability to characterize that phenomenon at the level of behavior. So here's the most common view, which is that fear is a mediator. So you have some conditioned stimulus or threat stimulus, doesn't have to be conditioned, but a threat stimulus elicits fear, and then the fear itself produces the freezing or the autonomic nervous system or the hormonal responses. So fear is the mediator. That is the standard textbook explanation. Here's the threat. Fear is there, and then you get responses. Now, William James long ago said, we don't run away from the bear because we're afraid. He was right. I don't think he was right in his explanation when he says we're afraid because we run. I don't think that as he said, the physiology of a fear response is such that it dictates what you feel. I think that it contributes to, but there's much more going on besides simply the body feedback that James and Antonio Damasio and others emphasize. But let's stick with the mediator role here. So my colleagues will talk about the conditioned fear, the frightened rat, the rat's frozen in fear. And that's what they mean when they say this, that fear is the reason the rat is freezing. Fear is frightening the rat. And the amygdala is often said to be how this fear comes about. So the stimulus activates the amygdala generates fear, so fear is there waiting to be activated in the amygdala, and then the rat freezes. But this is wrong. The amygdala is not the brain's fear center. No matter how many times you've heard otherwise, it's not. And I'm partly responsible for that view, so I feel qualified to tell you that it's not. This all gets into semantics, and the semantics happen to be very important, as I'll be explaining. We'll get to that. Let me just go on. So, what are the classic findings? So brain lesions. So the finding is Damage to the amygdala in animals or humans eliminates behavioral and physiological responses to threats. Okay, that's the finding. The conclusion, amygdala activation leads to fear, which causes responses, so amygdala damage must eliminate fear. Brain imaging. The finding is when people see threats, the amygdala is activated, okay? Conclusion, body responses to the threat must be mediated by fear in the amygdala. So it all comes back to this idea that, fear is in the amygdala. Surprise, a woman with amygdala damage still feels fear. How can that possibly happen? That's impossible because the amygdala is the brain's fear center. Well, that's only impossible if you believe that the amygdala is the brain's fear center. Headlines in Nature, Science, Discovery, Discover Magazine, Wired, Scientific American. Humans can feel terror even if they lack the brain's fear center. Scaring the fearless, on and on. Researchers scare fearless patients. So, what's really going on is the amygdala is a threat detector. It's detecting the threat and controlling responses to danger. It's not feeling fear. There's no fear bubbling up out of the amygdala when you detect the threat. This is something that rats do. Every animal that has an amygdala, this is what it does. And many animals, including all invertebrates, lack an amygdala completely. They have their own version of the amygdala, which is a circuit that detects and responds to threats. The ability to detect and respond to threat is not in the brain to make you feel anything. It's there to keep you alive. Bacterial cells can detect threats and move away from threats. So this ability is as old as life itself. It's a necessary component of being a living organism. You have to be able to detect what's harmful in your environment and protect yourself against it. Now, it just so happens that if you have a brain that can be conscious of its own activities, then you feel fear. But fear is not in any way intrinsically tied to this threat detection system. So, I've kind of said this already. Here's another finding. Children rated as anxious by their parents had a bigger amygdala. By the way, I'm not criticizing any of these studies. The studies are fine. It's just the way they're being interpreted, both sometimes by the scientists, but also by the media. Headlines, fear center in brain larger amongst anxious kids. Anxious children have bigger fear centers in brain, blah, blah, blah. So what's wrong with the headline? So behavior was rated. Anxiety was not measured. So the parents are rating their children on the basis of the behavioral responses of the children. Same way we would rate, you might rate a fear reaction in a rat if you were talking in that way, or an anxiety reaction in a rat. None of the children were clinically anxious. As I said, the amygdala is not the seat of fear, much less the seat of anxiety. If by fear and anxiety we mean conscious feelings. So, the other problem here is that, you have parents who themselves may be a bit anxious and will be interpreting their children in terms of their own anxiety about the children, and that's not factored into any of this. But the main point is that the amygdala is not responsible for anxiety or fear. Here's another one. Crayfish treated with a benzodiazepine were more exploratory, less behaviorally inhibited, in a chamber where they received electric shock. Headlines. Anxious crayfish can be treated like humans. Even crayfish get anxious. Crayfish may experience a form of anxiety. BBC was more tempered in their response. They may experience it. So you can see where things get out of hand. So now, we can use crayfish to test drugs like benzodiazepines. If the drug works in making the crayfish less behaviorally inhibited, in other words, less timid, more exploratory, then that should be a drug that will help people, according to this line of reasoning. But most of the tests done to develop new anti-anxiety drugs are done in rodents. And this work started in the 1960s, but after 40, 55 years or so of research on this, the big pharma are pulling out. So Andrew Whitty, CEO of GlaxoSmithKline in 2010 said, "We believe the probability of success is relatively low in finding new anti-anxiety agents." So the clinical outcomes of these effects have been disappointing, according to Weill and Holmes, two animal behavior researchers in this area studying this kind of stuff. As promising results with novel agents in rodent studies have been very rarely been translated into effectiveness in humans. So here are the tasks that are used to develop these drugs. And what they're doing is they're measuring behavioral responses to threats. How can you possibly measure a feeling of anxiety in an animal? Well, the logic is that if the rat is acting anxious, according to, this is like the parent judging the child, if the rat is acting anxious and a drug makes the rat act less anxious, then that drug should make people act less anxious and feel less anxiety. But the problem is you know, the president and CEO of GlaxoSmithKline said it doesn't work. These drugs, of course, they help to some extent, but they're not the panacea that you would expect after 55 years of research and gazillions of dollars. But the drug is actually doing probably exactly what it was designed to do, and in a sense, it's very successful. So, for example, if the drug makes a person with social anxiety more willing to go to the party, the drug is doing what it was doing in all these animal models and in the crayfish, making the person less behaviorally inhibited, even though when they're at the party, they still feel anxious. So, drug is a success. The companies just didn't know what they were trying to do when they did this. So, how do we account for this? Well, in 1984, before I did any research on rats on this topic, I wrote a kind of conceptual model of where I thought things were going. Now, I didn't come at this with a blank slate, really. I did my PhD working with Mike Gazzaniga studying split brain patients. And what we were interested in is how the left hemisphere, the talking, speaking left hemisphere of the patient would respond to behaviors that we generated out of the right hemisphere. So we would, do something to make the right hemisphere like scratch, the arm or wave or stand up, and then we'd ask the left hemisphere, "Why'd you do that?" Now the left hemisphere, of course, has no information about why the right hemisphere was doing that, but quick as a flash, the patient would come up with an explanation. So if he scratched his arm, he'd say, "Oh, my arm itched." If he stood up, he would say, "I needed to stretch." Or if he had waved, he said, "I thought I saw a friend out there." Or if the command was to laugh, he would say, "Oh, you guys are really funny." So, the idea that we came up with, and that Mike Gazzaniga has really focused on the rest of his career, is the idea that one of the jobs of consciousness is to interpret things that are coming out of systems that basically work non-consciously. Almost all of our brain is working non-consciously. As Freud said, that consciousness is the tip of the iceberg. And it's not non-conscious because of repression, the way Freud said. It's non-conscious because simply it's not conscious. There are no connections between the parts of the brain, systems in the brain that allow consciousness to come about, and most of the unconscious brain. I mean, your conscious mind doesn't need to know anything about the circuits in the brainstem that are controlling your respiratory rate, or the heartbeat, or regulating insulin release in your gut and things like that. So it's a good thing that we aren't conscious of everything. But sometimes, when you find yourself acting in a certain way, you need to kind of come to terms with this. What cognitive dissonance is all about. You have behaviors that are coming out of your brain for reasons you don't understand, so you consciously have to resolve that in some way and make sense out of it by making it part of your life story in some sense. So that's how I got to this idea that, that there are non-conscious systems in the brain that are controlling what we call emotional responses. And these systems produce these behavioral and physiological responses that we know and love as so-called emotional responses. But the conscious systems are involved that give us our feelings, such as fear and anxiety. These are subcortical circuits. For example, the amygdala is particularly involved in immediately present threats that we talked about at the very beginning, whereas the bed nucleus of the stria terminalis is more involved in future threats that are uncertain. There's a degree of uncertainty about when the threat will occur and whether it will occur at all. In that case, the bed nucleus is more important, but they work in sort of parallel at this subcortical level. Whereas the conscious feelings require the complex areas of prefrontal cortex, not just one little area. There are lots of areas of prefrontal cortex that are involved. The anti-anxiety drugs work on these subcortical circuits, but they're not necessarily working up here so well. You might say, well, what about benzodiazepines? They make people feel less anxious a bit, right? But two questions. One, do they really make people less anxious, or do they kind of just sort of numb a lot of emotional states? And two, it's also possible, and we know that there are benzodiazepine receptors here, so as well as here. So maybe they are making people less anxious because they're acting on both circuits. My point is not to, dismiss all drugs or to rationalize any particular drug, but instead to point out how we need to think about drugs and their effects on the processes that are important. And two of the processes that are really important, so one set is those subcortical systems that generate these responses. You walk out on the street and you jump back, the bus goes flying by, then you realize that you were in danger. A lot of our brain, a lot of our responses are controlled non-consciously like this, especially in the case of threats that are suddenly appearing. So we just need to separate these systems, because the threat is the source of activity in both of these systems, but it's having two different things. Now, in our conscious mind, we know the threat is there, the snake is at your feet, you see it, your heart is racing, and you begin to feel fear. How does that come about? Well, your amygdala has detected the snake, it's pumping out activity to the body. The amygdala is also going to activate cortical processes like the visual cortex. In the presence of a threat, the visual cortex is more highly activated than in the presence of a neutral stimulus. That's not because you have plasticity in the visual cortex that has encoded the threat so much as because the amygdala is activating the visual cortex. If you have a person who has amygdala damage, a threat does not give that boost of processing to the visual cortex. So what happens is the amygdala makes the visual cortex more active. That activity causes attention systems to scan the environment looking for the source of the threat. You have these scripts or schema stored in long-term memory. You see a snake fear. You begin to apply the fact that, okay, there's a snake. Snakes elicit fear. My body is revved up. I must feel afraid. I mean, that's how we get to these feelings. They're cognitive interpretations based on complex information. They're not simply encoded. The standard view is that fear is an innate emotion, that all these emotions, we have all these Ekman emotions that are innately programmed in our brain. I don't think that's true. These emotions are cognitively assembled in our minds. We have 37 words in English for different variations of fear and anxiety. We don't have 37 fear or anxiety circuits in our brain. There is no hardwired fear. Fear is something that happens when you put the pieces of the puzzle together in your conscious mind. And I'll talk more about that later. Well, this is kind of redundant with what I just said, but to put it in the William James way. So you see the bear, you have the conscious experience of fear, but the running is controlled by these non-conscious systems. So misunderstanding about the difference between threat detection and the feeling of fear and anxiety is widespread and is now built into the conceptual basis of the research, the funding mechanisms, ideas about what underlies uncontrollable feelings of fear and anxiety in the brain, uses of this research as a way to develop drugs to help people feel less fearful and anxious, and as the depiction of the work by the media. So it's pretty widespread. So why does this problem occur? Well, we're all anthropomorphic, and it's natural. It may even be an innate capacity of the human brain to be anthropomorphic. It's possible that it's beneficial to our species to treat animals as if they're little people, which is what we do. We all have pets, and I go home, I rub my cat's belly, and he purrs, and I treat him as if he's happy. And that makes me happy and makes him happy, and so we have a coexistence that works that way. But in the lab, I can't make that assumption, because there's no way I can measure my cat's fear or anxiety or happiness or anything else in his conscious mind. To conclude that an animal or even a person is having a conscious experience, you have to do two things. One, you have to collect evidence that's consistent with the interpretation that consciousness is involved. And two, you have to show that the phenomenon can't be explained in terms of non-conscious mechanisms. I would say that I don't know of any instance of a behavior in an animal that can't be explained in terms of non-conscious processes that somehow are controlling that behavior. It's not that I don't think that animals have some kind of experience. It's just that we're too free and too ready to attribute those things to animals and then use those as explanations of human behavior, when even in the human brain we don't need conscious fear to respond. So if my colleague Liz Phelps at NYU brings people into the laboratory and shows them scary pictures and measures their brain activity while doing this using fMRI, the amygdala will be activated, heart rate will increase, the palms will sweat, and so forth. If she shows the people the picture subliminally using various tricks that psychologists have, again, the amygdala is activated and the heart is beating, the palms are sweating. But the person doesn't know the stimulus is there, can't tell you what the stimulus is, and reports no feeling at all of being afraid or anything. So you don't need fear to have these responses. Fear is a separate process altogether. It's not the reason we respond. So have I covered all this? Let's see. Well, okay, so, Jane Goodall very confidently said, she's so revered I shouldn't be putting her down. But, animals show emotions similar to those we describe in ourselves. Animals feel pleasure, sadness, excitement, resentment, depression, fear, and pain. They're individuals in their own right. Now, these things don't necessarily go together. Of course, they're individuals. A bacterial cell is different from another bacterial cell, so it's an individual. So the fact that they're individuals doesn't mean that they have all this stuff. And how does she know what they feel? Again, this is an interpretation based on this idea that we know what's going on in another animal's mind because they're acting a similar way that we are. Even a dog that's injured and writhing in pain on the ground, we assume that dog is experiencing pain the way we do. It may be experiencing something, but what do we really know about its experience? And what we do know, though, is that a lot of these responses that will be exhibited by an animal that is been injured in this way are reflex responses and responses controlled by subcortical systems. There's not like this little thing in your mind, this pain, that is causing you to act in this way. Pain is another parallel process that you experience. A person, for example, who is in chronic pain, say chronic back pain, it's really just killing them all the time. If you tell them a joke and they laugh, momentarily they forget about the pain because it's been removed from attention by this other thing that's grabbed attention. These are conscious states that we experience when they make it into our attentive mind, into our conscious experiences. So, you know, we have to wear different hats when we're being a scientist and a layperson. Francis Bacon, who made this point long ago, he warned about the danger of using common language in science. We reify the referent of a word and give it a reality that it doesn't warrant when we do this. And this is exactly what happens when we use the word fear, which we have in our experience and from our own introspections, and apply that to another animal as if that animal has the same kind of mind and brain that we do. So here are a couple of quotes. Nico Tinbergen, the father of ethology, hunger, like anger, fear, and so forth, is a phenomenon that can only be known by way of introspection. When applied to another species, it's merely a guess about the possible nature of the animal's subjective state. Jerry Kagan, the esteemed developmental psychologist. Neuroscientists use fear to explain the empirical relation between two events. For example, rats freeze when they see a light previously associated with electric shock. Psychiatrists, psychologists, and most citizens, on the other hand, use fear to name a conscious experience of those who dislike driving over high bridges or encountering large spiders. These two uses suggest several fear states, each with its own genetics, incentives, physiological patterns, and behavioral profiles. The only thing I would quibble with is I would say that these are not two fear states. They're one fear state and a state of threat detection and response for the other. So I think I've probably covered a lot of this. I just want to emphasize we need to be careful not to confuse the correlation that goes on in our mind of the conscious experience of fear with the fact that our brain has also detected and processed a threat and responded to a threat. These things are fused together in time, and so our mind, our conscious mind, correlates them. But remember, correlation is not causation. We have to keep that separate. And it's only by taking these processes apart in the brain and following the flow of the stimulus from the sensory system into the subcortical brain to trigger these responses, and from the sensory system to the cortex and to the conscious brain to have these feelings. That we see that these are different things. You have to be able to take them apart in the brain to see their distinction, because they are so consciously, they're so fused in our conscious mind. We have this problem with our memories as well. So we have conscious memory or explicit memory or declarative memory, however you want to call it. And we also have implicit memory or non-declarative or procedural memory. And again, these were so long thought of as one, we thought there was one kind of memory in our brains. But through patient HM and through other studies, we see that the conscious memories are separate from the non-conscious memories. They were just so correlated, tightly correlated in experience, that they seem to be one kind of memory. So, as a neuroscientist, when I'm studying this so-called fear conditioning, what I'm studying is how the tone and shock come together on individual neurons, and those two stimuli meet up in the lateral amygdala. And that's where the association between them is formed. That association involves synaptic plasticity here between the tone and the shock. The shock is a strong stimulus, and so that strong stimulus creates certain chemical changes in this neuron that now makes it more receptive to this neuron, and it sort of glues in the connection between this neuron and the amygdala neuron. so that when the tone comes along later, it activates that neuron in a way that it couldn't do before, and then it flows through the system. Oops. This is called Hebbian or homosynaptic plasticity. And then we also have neuromodulators like norepinephrine that are released onto these neurons as well, and they boost that sort of extra glue that makes this connection more strong and more persistent. So, in describing how this kind of conditioning works, we don't need the concept of fear at all. As a scientist, all we need to understand is how that tone and shock come together and what sorts of changes take place to produce the responses that we're interested in. That's not saying that fear doesn't occur in the human brain or in some other animal brain, but it's not what is going on when we do this kind of conditioning process that produces these responses. So, this conflation of fear with the processes that detect and respond to danger has hijacked our understanding of these more fundamental processes and diminished our understanding of fear itself. We need a scientific view of how our organisms detect and respond to danger that recognizes the differences between these processes, can be used to study threat processing similarly in humans and other species without making unsubstantiated assumptions about conscious feelings in animals, and that can account for how these non-conscious processes contribute to human subjective experiences of fear and anxiety. So this is back to this picture that we saw earlier. Again, the anxiolytic drugs are acting on behaviors that are controlled through these circuits, whereas the patients feel fearful and anxious here. So one way to think about this, I told you that I view the feelings as this kind of construction in the conscious mind of a bunch of things. It's the coalescing of a lot of things at one time that bring together this feeling of fear. So we have sensory processing. There's a snake at your feet. These survival circuits like the amygdala and bed nucleus and so forth, subcortical circuits are activated by that sensory processing. The survival circuit is activating brain arousal, causing the conscious mind to now start seeking out an explanation. Attention gets focused to the environment, looking for the source of this arousal. This is the famous, in line with the famous Schachter and Singer experiments in the 1960s that you may know about. You get body feedback, as James and Damasio both argue strongly for, but it's only one piece of the puzzle. You get feedback from behavioral responses like proprioceptive feedback, but you also get hormonal feedback from cortisol, for example, goes back to the brain through the bloodstream. Epinephrine and norepinephrine are released into the circulation. They don't cross the blood-brain barrier, but they bind to receptors on the vagus nerve, and the vagus nerve then sends its connections into the nucleus of the solitary tract and the brainstem, which then connects to the locus ceruleus, and that further potentiates brain arousal indirectly. We have memory, different kinds of memory. For example, schematic memories of these schema that define different kinds of emotions. We have love schemas and fear schemas and disgust schemas and anger schemas and so forth. When some of the pieces begin to enter the conscious mind through a process of pattern completion, you have enough of the pieces of the puzzle, your mind coalesces on the conclusion that you're in fear. So we have episodic autobiographical memories, implicit memories as well. And then we have executive functions in working memory, attention, monitoring, labeling. You need monitoring to kind of absorb all this and begin to put it together. Labeling is part of what's coming out of the schema. Attributing, we not only, we sometimes have to guess about why we're having these explanations, about these experiences, and so we attribute meaning to them. So in a way, the working memory is kind of, the executive functions are sort of the spoon that stirs the pot. So what I'm talking about here is a kind of metaphor of making a soup. So soups are made not by using soup ingredients, but by using non-soup ingredients. water, onions, garlic, carrots, celery, salt, pepper, chicken, for example. Now, so you could take these ingredients, except for the chicken, and then maybe instead you would put shrimp in, so it begins to have a different kind of character. Or I'm from Louisiana, so we might keep the chicken in and maybe even keep the shrimp in, but then throw roux in there to give it, make it gumbo. So there are different ways that you can change the character of the soup by changing its ingredients. Salt and pepper are very important. They're kind of like arousal in the brain, because they don't change the character, but they raise the intensity or decrease the intensity of the flavor. And these are the things we've been talking about in working memory that work in the same way. Fear is not something that's built into the brain. There are no kind of emotion circuits in the brain, in a sense. There are circuits that do certain things, like keep you alive. These survival circuits, they're not there to make emotions. They're there to keep you alive. You know, we tend to associate fear with the defensive system of the brain. So when you see a snake or an angry person, the amygdala is activated, and that will produce a threat response, a defense response in us. But we can be afraid of a lot of things. You know, some other survival circuits are those involved in energy regulation and nutritional regulation, fluid balance, thermal regulation, reproduction, and so forth. These are all things that are necessary for the survival of bacterial cells and every organism between bacterial cells and humans. So the interesting thing is, each of those things can be a source of fear. Fear has no particular exclusive contract with the amygdala and the defense system. You can be afraid of starving to death, of dehydrating, of hypothermia, or too hot or too cold can kill you. You can be afraid of reproductive failure, and so forth. So fear is an experience that we put together on the basis of non-fear ingredients. These ingredients are general purpose things that are in the brain for other reasons. You know, we think of arousal as being especially important in emotions, but the arousal system is in the brain not to make emotions, but to regulate our level of alertness and sleep-wake cycles. It's very useful in a situation where something is dangerous to bump up the arousal, because that makes the system more efficient, because it sort of like recruits all of the brain to work on the particular problem, especially in a situation of danger. The brain gets monopolized, and its resources are being used for that. So, what are some of the implications for therapy? Well, we all know what exposure therapy is. So, you know, a common assumption in my field, which I only have to admit that I made this assumption myself for a long time, is that exposure therapy and extinction are the same thing. So people in my field will talk about studies in rats being extinguished to a tone that's been paired with a shock, saying that, well, this helps us understand exposure therapy, because exposure therapy is the way, extinction is what's going on in exposure therapy. And while that's certainly true, there is some extinction that goes on, and that's important because extinction is a well understood process in the brain. So information about the neural basis of extinction might be very important in understanding how exposure therapy works. But we have to be clear about what it is the animals can and can't tell us. And it's a kind of oversimplification on the part of people in my field that jump to the conclusion that if we understand extinction, we'll understand exposure therapy. You all know that that's not the case, because exposure therapy, let's talk about what happens in the laboratory when you do extinction. Well, first of all, you don't talk to the rat, obviously. You bring the rat in, you put him in the cage, and then you present tones one after the other till he stops freezing to the tone. Take him out. So it's all stimulus repetition. in human subjects studied in the laboratory, it's sort of similar. There's no stress management training, relaxation training. There's no cognitive coping skill training involved. You bring the subjects in and you give them stimulus repetitions and you weaken their conditioned response. But in exposure therapy, you do all those other things. I know there are different variations of it and different people will do different things, but there's much more going on than simply exposing the person to the stimulus in a repetitive way. So extinction can help us understand the repetitive aspect of this, but not the other stuff. So let's assume that I'm just going to make the assumption that extinction is relevant to exposure therapy and try to show you how it might be improved by things that we know about, which could then maybe improve exposure therapy to some extent. Well, so here's extinction. You condition the rat, and then you give the stimulus over and over, and he stops responding. So here's some of the key areas of the brain involved. Sensory systems bring the CS into the brain. The medial prefrontal cortex is involved in regulation of threat conditioning and extinction. The hippocampus and the contextual modulation of conditioning and extinction. And the amygdala in the acquisition and storage of the threat memory and of the memory of extinction. So through the sensory system you get the amygdala responses, and through the hippocampus you contextually regulate the amygdala, so a snake in the zoo is not as dangerous to you as a snake on a path in the woods. And the medial prefrontal cortex is what helps you kind of create a new form of learning that inhibits the amygdala and weakens the response. Extinction is inhibitory learning. It's not erasure of memory, it's inhibitory learning, creating a new memory that inhibits the amygdala's responses. So we can think of the prefrontal cortex as sort of the brakes and the amygdala as the accelerator in these responses, in these defense reactions. And we know, this is just, we know a lot about the circuitry within the amygdala, how the medial prefrontal cortex connects with it and all that. I'm not going to bore you with that. But here's, roughly where the medial prefrontal cortex is in the brain. So if we think of the brain as like a hot dog bun, we're looking at the two halves of the hot dog bun here. This is the brown toasty part of the left side of the bun. This is the white inside part of the right side of the bun. So this is showing where the medial or inside part of the prefrontal cortex is, and here's the lateral part. So the lateral part of the prefrontal cortex is key for working memory. Medial is also involved to some extent. But the medial is especially important in extinction. And this is a picture basically that I presented in Synaptic Self in 2002, where I made some mistakes about all this, where again I was equating extinction with exposure therapy. And now I'm trying to recant some of these mistakes. But one is that the fact is that extinction does require this connection between the medial prefrontal cortex and the amygdala, and it's a direct and strong connection. Whereas if you try to change the amygdala through working memory, ask the patient to think about what they're afraid of, and try to think about that not being so dangerous and so forth. There are no connections that will allow working memory to do that to the amygdala. They just don't exist. You got to go through these back roads and side streets, and that's why simply talking about a problem is a long, slow process. You're trying to create synaptic plasticity through stages to get to the amygdala through circuits that don't naturally exist. I mean, it's just a fluke of human evolution. This is the most recent acquisition in primate evolution, and it's just not wired up yet with the amygdala. Maybe there's some advantage to keep them separate, but there may also be the possibility that as we continue to evolve, these things are going to get better connected. I don't know. So as I said, exposure is much more than extinction. Extinction is stimulus repetition. Exposure, you have a lot of talk therapy and instruction, cognitive coping skills, relaxation trainings, and stimulus repetition. So we need to separate these processes to optimize extinction. Why do we need to improve extinction? Well, we know that you can extinguish someone completely, but this almost never happens. When you test later, you can get recovery from extinction. So the person is extinguished in the office and then goes out in the world and something scary happens and bam, it's undone. This is called, this would be a triggered form of, it could be by a trigger stimulant, but simply the passage of time, as Pavlov discovered, is enough to cause the spontaneous recovery of extinction. So how can we make extinction less recoverable, in a sense? So here's some ideas. One, extinguish in multiple contexts. Because learning is context dependent, the effects of exposure therapy don't always carry over into real life. So if you do exposure in many different contexts, it might affect might make the effects more general and more persistent. Maybe focus on core triggers one at a time rather than the whole memory. Memories are very complicated, and we can't extinguish the memory of a trauma. We have to take the triggers and extinguish them because there's too much going on. In the process of extinguishing one part of the trauma, you may facilitate another part. Space versus massed learning. We know we were all students at one point, we're students throughout our lives, and we know that when you cram, it's less effective as a way of learning than when you space the learning out over time. And there's a reason for this, and it's because there's an enzyme in the brain called CREB, you don't need to know what that means, but that enzyme gets depleted when you cram, when you have too much information in one session, one small segment of time. And then it takes several hours for the CREB to replenish itself after you stop cramming. So if you had longer sessions, now we're not talking about practicality here and what insurance companies will pay for. What I'm talking about is, in an ideal world, what could you do to a therapy session that might make it better? Well, making it longer so that you could have more time, more spacing between groups of exposure trials, for example. Reduce interference. So as soon as you learn something, anything that happens right after that can compete with circuits that are storing the memory and interfere with it. So after a therapy session, ideally, you would have a place where the person could be engaged in activities that would be memory promoting rather than memory interfering, letting that memory begin to consolidate and strengthen as time goes by. But we also know that memories consolidate best during sleep. So you would also have a sleep room where the patients could then sleep for several hours. Or maybe all of this would be done as an outcall in the person's home. So they could go right to sleep in a safe environment where this is where they would feel comfortable. I did find one study that showed that CBT was more effective after a nap. So this was not this was in the laboratory. So they did CBT and then had a nap area, and the patients took a appliance. I'm not sure what you call them, but took a little nap where they actually went to sleep, and those that slept had more effective, persistent effects of the therapy. Now, we can combine drugs with extinction as well. This isn't drug therapy. This is simply kind of cognitive enhancement of extinction, or pharmaceutical enhancement of extinction. So drugs like D-cycloserine you've probably heard about, which were discovered by Michael Davis as being able to facilitate extinction in rats, and therefore he started a clinical trial in people, and there's been some success. I think it hasn't been completely successful. It hasn't been replicated in every possible study, but there seem to be promising results, at least in some studies. So there are many other drugs that we know affect learning, and these could be tested in rats and then perhaps in people as well if they have an effect. Non-conscious extinction. Now, if the conscious mind and the non-conscious mind are extinguished simultaneously, as would be done in a typical exposure therapy session, it's possible that you're going to create competition between brain areas that don't play together well. And the conscious mind will begin to suck up, it's more powerful in a way, and it will begin to suck up resources and make the non-conscious learning less effective. So what you could do is, if you had, this would only work in the case where you have specific, like visual triggers that are well-defined and so forth, for example, in a phobic situation, where you could then do masking presentations, masking extinction, where the person doesn't know the stimulus is there, but yet the brain is being extinguished, the amygdala is being extinguished non-consciously. So you don't have, the conscious mind can be doing something else altogether, thinking about, you could give it some task to do and let the non-conscious mind do its thing there. We're actually doing a study at NYU with Liz Phelps on this, not in patients or clients, but in so-called healthy NYU undergraduates, to see if we can get effective non-conscious extinction. And I don't have results yet, but I'm very optimistic that this is going to work. There's actually a study done by a researcher at SUNY New Paltz, not New Paltz, what's the one in Westchester? Purchase, yes, Purchase, thank you. That looked at spider phobics who were given a single exposure to a picture of a spider, either consciously or non-consciously. And then a year later, they measured the avoidant behavior of the people, or asked them about their feelings about the stimulus. In the case, and these were conscious presentations. So in the case where the people had the non-conscious exposure, they exhibited less avoidant behavior, but still felt anxious. Those that had the conscious exposure were less anxious, but they still avoided. Now, I don't know how much weight to put on this. It was, one exposure. How can one exposure be that powerful? But, it seems to be a good study, but it definitely needs to be replicated. But this was an inspiration for me to begin to pursue this idea that maybe non-conscious exposure would be an effective way to kind of get the brain, reduce the arousal that the amygdala is going to generate every time you're exposed to that stimulus. Because you kind of, weaken the amygdala so that when the phobic stimulus is there, it's not freaking the person out consciously, because it's not the person not getting all this body feedback and brain arousal that would then cause them to be seeking an explanation for why they're experiencing all this. Now, so this might be a sequence that you could follow. First, you do non-conscious extinction. Then once you've got the arousal and all that taken care of, you then do conscious extinction. And then maybe you want to do some belief change and talk therapy to get at, the thoughts and schema and so forth that are kind of keeping the conscious mind looking for those triggers and assembling these states as well. Now, the final thing I want to talk about is changing or erasing memories. We can make memory of trigger stimuli inaccessible using procedures called reconsolidation. And this works very well in rats. It's still being tested in humans. That target specific triggers. Or we can achieve the same goal by adjusting the timing of stimulus exposures during extinction. I'll tell you about that in a minute. The latter approach is is very powerful and has been shown to actually reduce craving in addicts. So I'll tell you about that study. This wasn't done by us, but it was following up on a study a couple of studies that we did, one in rats that was done by Marie Morfield, and one by Daniela Schiller in Liz Phelps that was done in humans. So through a mistake in Marie's animal studies, she had a space put between the first exposure trial and the rest of the trials of exposure in rats. So it was about a 10-minute space. Usually it would be one every minute or so. So she did one 10-minute wait, and then did the rest over and over again. And the rats extinguished. And when she did that, she was testing spontaneous recovery and triggered extinction, triggered recovery and all these other things. And there was never any recovery. The memory didn't come back at all. So she then did a study where she compared directly the effects of doing the same number of exposure trials, either all together or in this spaced out way, and keeping the timing of the session the same by adjusting all this stuff. So it was a very well-controlled study. And the rats that had the regular extinction always recovered their problems, whereas those that had the spaced training, the space between the first and second trial, never recovered. So then Daniela and Liz did the same thing in human subjects, and with conditioning in the laboratory, so with visual stimuli and shocks again. So the people never recovered the response after conditioning. We brought them back several times. Submitted the paper to Nature. They said, well, how do you know it won't come back? Bring them back after a year. So we waited a year, brought them back, and there was no recovery. So they published the paper. So then these guys, a group in China and at NIH, said, well, let's try this in addicts. And so they did exposure to relapse cues to reduce the cravings, which will work to a certain extent, but then they always relapse at some point. And they did it that way, or they did it our way with the 10-minute break between the first and second trial, and then give the rest of the trial. So they did it in rats, and the rats relapsed the first way, but didn't relapse our way when they were given the relapse cues. So they did it in cocaine addicts and got the same thing, a reduction in relapse by spacing, putting a 10-minute space between the first trial and the second trial of exposure. So these little things that we play around with in rats can have profound implications for what happens in the human brain. And I could go on and talk about reconsolidation, but I think I'll save that for another time, because this is much more exciting. It doesn't require any drugs. It's just a matter of spacing the first trial and the second trial of exposure. So let me skip all that. I'll just say goodbye. Thank you.
Do we have some questions?
Over here.
Hi, thank you. This is wonderful. Is the last thing that we're talking about, is that kind of that space for the mass?
No.
No, it's not.
I mean, it could have something to do, I don't think that's the answer. So to understand that, we kind of need to talk about reconsolidation a little bit. So let me just say a brief word about it.
Okay, so the traditional view of the way memories are formed is we learn, we store the information, then when we retrieve what we stored the first time. When we retrieve later, again, we retrieve what we stored the first time. So there's like an engram that that gets created, and then every time we remember something, we pull information out of that engram. Again, kind of this was a study that was done by someone else in my lab, Kareem Nader, in 2000. And I actually told him not to do it because I didn't think it would work. But he was a smart guy and he went ahead and did it anyway. We've been studying the effects of protein synthesis inhibition on the consolidation of memory. So if you give a protein synthesis inhibitor, after learning, you get short-term memory, but you prevent long-term memory from being formed. Because in order to form long-term memories, neurons have to synthesize new proteins that then glue the connections together. So he had found this research showing that if you block protein synthesis after retrieval, you could also disrupt memory, which was bizarre. And that's why I said I didn't think it would work. Why would that possibly work? And so, you know, the twist that we had when we were doing all this work was that almost all of the previous work had been done by giving the drug systemically. So if you give a protein synthesis inhibitor to an organism, person, or a rat systemically, it will make them very sick because you're shutting down protein synthesis, which is necessary for the activity of every cell in your body. So it's not a good thing to do, and you certainly couldn't do that in a human. So what we did was instead of giving it systemically, we put the protein synthesis inhibitor in the lateral nucleus of the amygdala where we knew the memory was being formed. And that required such a tiny, small amount that stayed right there in that little piece of the brain that didn't get into the body and didn't have all these other systemic effects. So we're able to block long-term memory storage by injecting the protein synthesis inhibitor into the lateral amygdala after learning. And then we could block memory after retrieval by injecting protein synthesis inhibitors in the lateral amygdala after giving the rat the tone, after they had fully consolidated the memory. So that led to a different view of memory. So we learn, we store, we retrieve, and when we retrieve the next time, we retrieve what we stored the last time. In other words, after each retrieval, you have to restore. So the idea is that retrieval destabilizes the memory to some extent, if there's new information available, and allows you to update it. This is not a way to disrupt memory. This is a way to update memory, because every time you remember something, you have to add contextual nuances to it, or you may have to even completely change what you thought was involved. So when you retrieve, you have to restore the memory in order to keep it. And if you block that, it's a kind of quirk of the system that if you block the restorage, you lose the memory. That almost never happens in nature. Almost always what happens is you update the memory when you retrieve it. So when you retrieve, you update, and then when you retrieve the next time, you retrieve that updated or new memory that you stored. So that's what we were doing. So this process of reconsolidation, let's see if I can give you a hint. Okay. So basically, the idea that we ended up with is that when you store the memory, then you retrieve it, you can alter that memory in two ways. One, by giving a drug. For example, the beta-adrenergic antagonist propranolol, that's used for hypertension, but also for stage fright and other things, because it acts in the body. But it also goes to the brain. It acts in the brain to reduce norepinephrine released, which is a major source of brain arousal. And brain arousal and norepinephrine are very important in the protein synthesis process and the stabilization of the memories. So by blocking norepinephrine in the brain, you can impair the memory storage process just as you would if you blocked protein synthesis. It's not quite as strong, but it does work pretty effectively. So to do that, you have to inject the drug after retrieval. You have a 4-hour window in order to do that. If you wait longer than 4 hours and inject the drug, it has no effect. So the question that we accidentally asked in that study where we spaced the training out was, what happens if you retrieve the memory and then do extinction? And what we found was that when you do that, you restore the memory as safe. The tone is now a safety signal rather than a threat. Because during that 4-hour window, you've created the memory that it's safe by doing extinction. And then when you retrieve it later, you no longer respond to it. So the 10-minute window, you may be asking, why do you need that 10 minutes between the first and second trial of retrieval? Because this reconsolidation process takes about 10 minutes to kick in molecularly. If you do it if you do it at 3 minutes, it doesn't work. You gotta wait at least 10 minutes to let that reconsolidation process kick in, then you can extinguish, and then you have a safe memory rather than a dangerous memory. So that's what's going on there. Good. Yeah.
Why wouldn't that kick? So let's say you do 20 trials, all spaced in a minute, right? But that last trial happens, and then nothing else happens for 10 minutes, and the experiment's over. So you've got your reconsolidation. Why would they then have a kick back later on? Like, why wouldn't that happen anytime the last learning trial is a safety trial and nothing else?
Good question, and I think the answer is because these processes are triggered by the stimulus. So if you in fact we can do this, if you give one reconditioning trial with the shock again, then you've wiped it out, and that's what gets carried forward. So if you do extinction, and then the rat is removed from the environment and nothing else happens, or even if it stays in the environment but nothing else happens, then it's still, it gets consolidated that way. I'm making that up, but the fact is that's how it works. Yeah.
By retrieval, are you saying that in that 10-minute gap before you begin the extinction trials, which in the simplest model would be, okay, so the unconditioned stimulus would be a shock, the conditioned stimulus would be a light, so you pair that a bunch of times, next day you come back. Are you saying then you actually go ahead and give the shock with the light, then wait 10 minutes, then do just the light, the light, the light?
No, just the light once,
Yeah.
wait 10 minutes, and then,
You don't need the shock, that first one, just the light.
no, the shock would condition it more strongly. You'd have to start over again the next day.
Just thinking if fear is a function of cognitive processes, that's what you were saying before, just to clarify. Are interventions such as like biofeedback and neurofeedback then just as flawed as benzodiazepines that are targeting the amygdala and these behavioral physiological responses?
I think,
So the question is whether the processes that would target the amygdala are flawed as a therapeutic approach. I think you have to do both. That's why I say you start with the unconscious extinction. I guess that would be maybe kind of like biofeedback or what else is it?
Neurofeedback.
Neurofeedback. So that would be like kind of trying to do what the unconscious extinction would be doing, which is kind of reducing what the amygdala is going to do to mess up the therapy that you want to do on the cortex. But I don't know enough about biofeedback and neurofeedback to know if that's what they really do. Yeah.
So I'm just fascinated with you that it was just 10 minutes and it made such a difference.
Right.
And I'm wondering if 10 minutes is just like this magic number, or has it been done with 20 minutes, 30 minutes? Is there like an optimal amount?
Yeah, we don't know. 10 minutes, less than 10 minutes doesn't work, but we don't know. We don't know the outside limit.
So it might work even better if it's like 30 minutes.
Might
But it
be.
might not work at all.
Right, yeah. And we should do that, but we haven't done that. Well, yes.
Hi, thanks for a great talk. I have two very unrelated questions. One is if you're at all familiar with the process, which is sort of involves desensitization, which is called EMDR. And the other is any age-related differences in the reconsolidation process.
Age-related?
Right, age-related in terms of working with clients of all ages.
You know, I know what EMDR is.
Okay.
I don't know enough about it to
Okay.
say anything meaningful. And I don't know if there are any age differences in reconsolidation. As far as I know, it hasn't been studied in elderly people. There could be.
Okay.
And you know, the other thing to point out is that these processes happen in different systems. So you can get reconsolidation in the hippocampal system, and also in the amygdala, and those are different. So I would think that in the aging brain, maybe the hippocampal system may be more susceptible to problems in later life in the amygdala. But I don't know.
Okay.
Question regarding the non-conscious distinction in the clinical setting. Maybe I missed something, but can you give me an example of whether it's a patient with fear of dying or fear of remaining single?
You would need something that's more discreet.
Yeah.
You know, with these procedures, you're using very brief exposures, followed by masking stimulus that prevents the stimulus from entering consciousness. So I'm not sure how you would get fear of dying into that scenario. So, you know, snake, spider, elevator picture, things that are strained by physical objects, things that can be controlled.
I have some colleagues who've been doing some research and application based in this reconsolidation model, but rather than beta blocker or another extinction trial, it involves a compassion focused CBT approach. So there's a combination of heart rate variability training, imagery that evokes an affiliative emotional response and sort of activation of oxytocin vasopressin system. And I'm wondering if that seems like a reasonably consistent with the theory that in that interval.
Anything that you do therapeutically in that interval should be working better.
No, okay, good.
4 hours, yeah, 4 hours. Or 3 hours.
I was intrigued by the idea, because I'm thinking of veterans, and anger is activation of the amygdala, walk into a bar, could be a fight. And then they're in jail, and the whole thing is undone. So working on the amygdala first, maybe by presenting them with subliminal pictures, along with that anger management and all the other things we do, before you begin the prolonged exposure therapy and so on, might be more effective because
Well, that's
it'll
the
keep them out of
hypothesis.
trouble.
Right.
If we can get that response, that activation reduced.
Yeah, that's the basic idea. So somebody tries it.
So I have a question about the more clinical application of this. For example, a soldier that just experienced an encounter that could lead to PTSD, or just someone that just experienced a traumatic moment. Could we somehow intervene and within a 4-hour period find a way to inject the drug into the person so that they don't encode the memory as traumatic as it was and prevent them from all the emotional and trouble that they'll go through afterwards?
Yeah, I mean, that's the idea. And, it's hard to evaluate the studies that have been done because they're usually very complicated. I think that these things are going to work best on really simple components, as I said earlier, the traumatic response, a very simple trigger as part of the trauma, rather than the big trauma itself. And you know, I understand the practical limitations of that. You know, the patient wants it all to go away, not just that little thing. Right? So maybe they go along with that. You know. My job is just to throw ideas out. So I want to just do, before we go, as Dennis knows, I'm also a musician, and we talked about maybe having some music here, but we didn't get around to that. But I brought a video that we can look at. The music I do is related to all of this stuff. It's about mind and brain and mental disorders. If I can find it. And this was done a long time ago, so it's not completely kosher with my new views, but I'll let it play anyway.
otherwise music. Buy my book, Anxious. Sorry?